Let's go to the minus polarity single-stranded RNAs. There are a number of important human viruses here. The paramyxoviruses, including measles virus and mumps virus, important childhood infections, completely controllable by vaccination. Rhabdoviridae, these contain rabies virus, the infection transmitted by rabid dogs. The filoviruses, everyone should know about the filoviruses now because there was just a big outbreak of Ebola virus infection in Western Africa. The filoviruses are unusual filamentous particles. They include not only the Ebola viruses, but Marburg viruses, first discovered in Germany at a primate facility. The orthomyxoviridae includes the very important human pathogen, influenza virus, which causes annual outbreaks of respiratory disease in many, many millions of people. And finally, the arenaviridae, of which Lassa virus is the best known. This is a virus that also causes a hemorrhagic fever, largely in Africa. These viruses have a negative sense RNA genome. And from what I've told you so far, you should be able to predict exactly what is going to happen in this flow of genetic information. Minus strand RNA cannot be translated, and the cell cannot make a copy of it to make messenger RNA. So you can predict the answer to the question, do negative-stranded RNA viruses carry an RNA polymerase in the particle? The answer, of course, is yes, because it has to come in with the RNA and then copy it to make plus-stranded RNA. If there were no polymerase in the particle, that would be the end of the infection because the negative RNA can't be translated. It can't be copied by any cell enzyme. That would be the end of the infection. So these genomes have evolved to include the RNA polymerase in the particle. So plus-stranded RNA can be made, messenger RNA can be translated into proteins, and also plus-stranded RNAs can be made to make more genomes, to make negative RNA genomes, which get incorporated into the particles. There are two kinds of viruses in general with negative-stranded RNA genomes. We have some, like the influenza viruses, where the genome is segmented. It's in pieces like the double-stranded RNA genomes of the reoviruses. So you can see here, there are 8 negative-strand RNA segments in the influenza viruses. Then there's another kind of negative-strand RNA virus where the genome is not segmented. It exists in the virus particle as a long RNA for the paramyxoviruses, measles and mumps, 15 to 16 kilobases. For the rabies-like viruses, 13 to 16 kilobases. Again, these are all negative-strand viruses, 2 general ways of assembling them in the virus particle. Having a segmented genome, the influenza viruses, the reoviruses, and many others, is very important for genetic variability because of this example. When you infect a cell with one of these viruses, the RNAs are replicated in the cell, and you have many, many copies of the RNA, which are then packaged into new virions. If you would, by chance, infect a cell with 2 different viruses, here we're showing 2 different influenza viruses labeled L and M infecting the same cell. If they both infect a cell, their RNAs are all going to be replicated at the same time. They're all mixed together. And the new viruses that are produced are not only going to be L and M viruses, but recombinants that have RNA segments from either parent. And one of the recombinants is shown here. It's called R3. And you can see that second RNA segment is red. It derives from the red parent, and the rest of them are from the blue parent. In fact, you could get almost any reassortant possible out of this co-infection, and this is partly the reason why influenza viruses are problematic. They vary extensively, and this is one of the reasons, and this kind of variation by reassortment is why we generate new influenza viruses on a daily basis, and why every 30 to 40 years we have a brand new strain that causes a global pandemic, because it's a reassortant caused by this process.